Hello everyone, I am Dr. Prarthana, Assistant Professor from the Department of Public Health Dentistry, Vinayaka Mission Sankaracharya Dental College, Salem. Today we will go into clinical trials. So it is something related to experimental design of epidemiology. So coming to clinical trials, we will today have an insight about introduction, types of clinical trial, design of clinical trial, statistical consideration and ethical consideration of a clinical trial, conclusion and some discussion. So coming to the introduction, clinical trial is an experiment or a quasi-experiment that tests the hypothesis concerning the effect of intervention technique applied to the individuals, or in simple terms, it is a planned experiment on a human being which is designed to evaluate either the effectiveness of one or more forms of treatments. So it's a planned experiment on a human being. So clinical trial is a part of experimental or interventional studies, as I have already mentioned, and it's a deliberate application or a withdrawal of suspected cause, or we can change one variable in the causation chain in the experiment group which will make changes in control group. So aim of clinical trial is to provide a scientific proof of etiology or the risk factor of various disease which may permit the modification or control of those disease, to provide a method of measuring the effectiveness and efficacy of health service for the prevention, control and treatment of disease, and thereby we improve the health of community. So going back to the history of clinical trial, to be precise, it is a history of health and disease. So in terms of public health, earliest time 1747, James Linn's study on scurvy patient, he found in a naval ship that vitamin C intake patients had reduced scurvy when compared to others. So some people in this navy ship who had eaten the rats and also who had the supplements of vitamin C rich fruits has reduced scurvy patients. So in 1796, Edward Jenner experimented with the cowpox. He injected this cowpox into a small 8-year child, and he found this cowpox has an immunity against smallpox. So this was the breakthrough in the public health history. And in 1920s, the methodology of clinical trial was, which we already know, who had been involved in the streptomycin trial, and he became the famous landmark of collaboration with Richard Doll on the correlation between smoking and lung cancer. And in 1953, first randomized control trial on tuberculosis patient was conducted. So coming to the types of clinical trials, we have various types based on various factors. So first we have explanatory trials and pragmatic trial. Explanatory trials where main purpose is to provide the biological information, and pragmatic trial which aims to test the procedure under the condition in which they would be applied in practice. So based on the control group, we have uncontrolled and controlled trials. Uncontrolled trial where if the design of trial features has no concurrent comparison, but rather they have an implicit or inside or a historical control. In control trial, we have at least one concurrent comparison. It can be either randomized or non-randomized. Depending on the randomization, we have randomized clinical trial and non-randomized or non-experiment or quasi-experiment. In randomized clinical trial, where randomization is used for allocation. And in non-randomized trial, those departing from strict randomized practice in such a manner that non-randomization does not seriously affect the theoretical basis of the conclusion. So for example, we have natural experiments, water fluoridation study. It definitely doesn't need any randomization where it will not affect the result. In such cases, non-randomization can be applied. So based on the study design, it can be concurrent parallel design, where comparison made between two randomly assigned groups, one group being exposed to specific treatment and the other not being exposed to the other treatment. Advantage is that it is simple in execution and implementation. So parallel comparison requires information about the subjects before as well as after interventions. Accounts for possible confounding factors and modifiers for the analysis. And then externally controlled trials. Here we will find the findings from the experimental groups are compared with the data obtained from the other sources. So may be used for ethical reasons. We find for an external control, for example the historical controls. Disadvantage is that it may differ in the way of treatment is done and recorded, and results may be misleading and they make subsequent RCT unacceptable. And sometimes it provides the clue for the medical sciences. Self-control trials use subjects as their own control and thereby reducing the confounding factor. So for example, we have before and after studies where comparing the incidence of disease before and after introduction of a prevention or curative measures. So that is an example of self-control trial. Then crossover trial. So this is an upcoming field where a subject is given the different treatments under comparison one after the other. So the treatment comparison will rely on the variability within and not between the patient. A washout period is definitely required to switch between the treatments. So this method is not feasible for the treatment with proactive washout effects or carryover effect, and this patient is successively given both the treatment and the placebo and the effect is noted. So they will given be, for example, group A and group B. Group A is given treatment A and group B is given treatment B, and over a period of time they are subjected for effect, and after that a given washout period of 1 week, for example. After this washout period, they are switched between the treatment, where group A will be given treatment B and group B will be given treatment A, and the effect will be noted. So this is a crossover trial. Split mouth design. Dental arches, quadrants, sectants, for examples within the patients, they are randomized for the treatment, not the patients are randomized. So this is very useful when the sample size is reduced and not applicable for systemic therapies where it can affect both the split mouth designs. And recent evidence indicate the crossover effect may occur in this design. This may obscure the true effect of the therapy. So we have split mouth parallel design. So for example here, patient 1, patient 2, patient 3 and patient 4. In patient 1, area 1 and area 2, treatment 1 and 2 is given. Similarly, in patient 2, treatment 2 and 1 is given. So they have equally given treatment 1 and 2 in the one side and the other side. There is no crossover between. So it is a parallel split mouth design. So coming to formal experimental design, we have other experimental designs which is completely randomized block designs, Latin square and factorial designs. So completely randomized design. It involves two principles: principle of replication, principle of randomization. Two forms: two group simple randomized design and random replicate design. Here two group simple randomized design is usually followed, where the population is defined first and then from the population a sample is selected randomly. After being selected randomly from the population, the sample is randomly assigned to the experiment and the control group. The limitation is that individual difference among these conducting the experiments are not eliminated. In replicate design, the limitation of this is two group randomization design is usually eliminated within the random replicate design, and it provides control for the differential effects of extraneous independent variables. It randomizes any individual differences among those conducting the treatment. Coming to randomized block design, where it is applied in heterogeneous group. In this design, subjects are first divided into groups known as blocks, such that within each group the subjects are relatively homogeneous in respect to some selected variable. So for example, if a group is heterogeneous, for example male and female are there in a single group, they have to be first separated according to the gender, male separate group and female separate group. So two different blocks are separated, where within the block they are homogeneous, and the number of subjects in given block would be equal to number of treatment, and one subject in one block will be given randomly one treatment. So main feature is that each treatment appears the same number of time in each blocks, and this design is analyzed by two-way analysis of variance. Latin square design is very frequently used in agricultural research. For example, if an experiment has to be done on 5 fertilizer used for agriculture, there will be varying fertility of the soil also. So two factors are involved here. Here the soil fertility is also involved. So this design is used when there are two major extraneous factors such as varying soil fertility and varying seeds. So it will enable the difference in fertility gradients in the field to be eliminated in comparison to the effects of different varieties of fertilizers on the yield of the crops. So limitation, it requires number of row and column, and it is very complicated box, and there will be considerable difference in the row and column. And the step ahead of this is factorial design, where it is used in where effects of varying more than one factor are to be determined. It will be simple and complex. For example, here the purpose of the experiment is to examine how different combinations of alcohol and caffeine affect response time in a stimulated emergency diving situation. So here, for example, in box 1, the responses time scores for a group of participants who received no alcohol and 0 mg dose of caffeine. Similarly, for second group, responses of time scores for a group of participants who received no alcohol and 200 mg of dose of caffeine. So in this way, separate factors are determined initially. So this is factorial design. Based on the objective, the clinical trial is again divided into 4 types. First, phase 1 trial. It is usually proceeded as considerable in research. It is in vitro or animal design. The purpose is to establish a preliminary evaluation of safety and first line outline of pharmaceutical or pharmacokinetic and where possible a pharmacodynamic profile of active ingredients in human. So phase 1 trial are typically in a very small groups, and phase 2 trial, it is done on volunteers selected on strict criteria, usually done in 100 to 300 subjects, and it assesses the effectiveness of the drug and appropriate dose safety. So initially phase 1 trial is an in vitro design and phase 2 trial is a safety design, and phase 3 trial is a clinical trial appropriately and performed on patients who give consent selected after strict inclusion and exclusion criteria, and it is usually done in large group of people like around 1000 to 3000 people. It will assess the effectiveness and safety and continued use of the drug in larger and more heterogeneous population, and it is carried out in a hospital patient, may also be done in outpatient in an intense follow-up. Phase 4 trial collect the information or long-term data on morbidity and the late effects in the large population. It will reassess the continual use of the drug and effectiveness and safety and acceptability. It is usually done under conditions close to the normal as possible. So based on the use of clinical trial, it is again divided into preventive trial. Preventive trial implies a primary preventive measures. So analysis of preventive trial should clearly state the result in terms of benefit to the community, risk involved and cost of the health service. Risk factor trial, where it involves trial of risk factor in which the investigator intervenes to interrupt the usual sequence. So in other words, we will remove the risk factor or intervene the risk factor. For example, major risk factor for cardiovascular disease is elevated blood cholesterol, smoking, hypertension and sedentary habit. So we can either intervene to stop them or modify them or give them any treatment. So that is risk factor trial. Cessation experiment. We attempt to evaluate the termination of an habit that will lead to a considerable disease. For example, smoking and lung cancer. So trial of etiological agent, it aims to confirm or refute an etiological hypothesis. For example, development of retrolental fibroplasia in premature babies exposed to oxygen. Evaluation of health service, to assess the effectiveness of health service, set up a priority for implementation of large number of activities which could contribute to the welfare of the society. So how will you design a trial? A clinical trial designing, it will involve protocol writing, selecting reference and experimental population, make necessary exclusion and inclusion criteria, then randomize the selected participants into experimental group and control group and manipulate or intervene and then assess them. So coming to protocol, is a scientific planning of document for the trial. So we need an appropriate plan to continue with the trial. So the protocol should be dated and signed by the investigator and the institution involved and the sponsor details. Objective is to assist the investigator to understand the research, provides a sounding board for external comment and orients the staff for preparation of forms and data processing procedures. So what are the content to be included in the trial? First, protocol title, identification number and the date, the name and address of sponsorer and monitor, name and title of investigator who is responsible for this trial and the address, telephone number for the communication, name, title, address and telephone number of qualified physician responsible for this all trial site related medical decisions, and then the clinical laboratory details and other medical technical departments involved in this trial. These are the general information necessary. And then coming to the background information, investigator product name and description, summary of findings, summary of known and potential risk and benefit, description and justification for the route of administration, dosage and treatment period. Statement that the trial will be conducted in compliance with the GCP guidelines, which is good clinical protocol, and description of population to be studied and reference of literature relevant to this trial. So trial design and objective to be specified. Trial design, either description of the type and design of the trial to be conducted and description of measures taken. For example, randomization and blinding for reducing the bias, and the trial objectives, investigators should define and reflect the specific objective in terms of quantifiable endpoints, and mostly the study objective can be summarized in the form of null hypothesis always. So recently regulatory agents have classified this trial objective into efficacy claim, superiority claim, equivalence claim. Efficacy claim must be tested in placebo controlled trial. Superiority claim are dictated design with inclusion of positive control receiving a standard surgical or non-surgical therapy. Equivalent claim based on either lower toxicity or financial cost compared to conventional treatments. So on this basis you have to select your controls. Selection and withdrawal of subjects. So inclusion and exclusion criteria should be mentioned properly, when and how to withdraw subjects, type and timing of data to be collected, how subjects are replaced, follow-up of the withdrawn subjects. So treatment of subjects, the treatment to be administered should be specified clearly, procedure for the monitoring subjects should be mentioned, and assessment of efficacy, specify the efficacy parameters and methods and timing for assessment. For safety assessment trials, specify the safety parameters, methods and timing for assessment, recording analysis of this parameter, type and duration of follow-up. So statistical analysis also had to be taken into consideration, where what is the method to be employed, sample size determination and calculation should be mentioned, level of significance should be specified, criteria for terminating the trial should be mentioned, procedure for accounting for missing, unused and spurious data has to be mentioned, procedure for deviation of plan, statistical plan, quality control and quality assurance, ethical consideration, data handling and record keeping, finance and insurance, publication policy has to be mentioned clearly. And always get an informed consent in a clinical trial. Dentist must obtain the individual's free and informed consent before his participant into the trial, based on two principles: personal inviolability and free will. From a legal point of view, consent is nothing but the patient accepts through an equivocal act of will to assume certain obligation. To be valid, the document must include two parts: a transcription of the oral explanation, a signature of adequate consent form. So it is nothing but the information sheet and the consent sheet. Institutional review board or independent ethical committee has to be given a certificate. Safeguards the rights, safety and well-being of all trial subjects. The composition of this team will include 5 members. At least one member whose primary area of interest is non-scientific area, and the other member who is independent of institution or trial site. So documents should be submitted to this IEC or IRC, will be trial protocol with amendment, written informed consent form, subject requirement procedure, written information provided to the subject, the investigator brochure, available safety information, information about the payment and compensation. Coming to the selection of population, we have to select a reference target population or sample. It is the population to which the finding of trial is found successful or expected to apply. Experimental or study population, where actual population that participate in the experimental study, should be randomly chosen from the reference population, and it should be representative of the reference population. So we should have two groups. We have experimental group and a control group. And then randomization is always necessary in a clinical trial if it is randomized, and if it is not randomized, it is not actually necessary. It is a statistical procedure by which the participants are allocated into the groups like study and control group. It is done to eliminate the bias and allow for comparability. Ensures that investigator has no control over the allocation, so it will eliminate the selection bias. So the patient randomization will be done usually by random number table, coin method and random permuted blocks. So it will ensure a balanced randomization, for example 1:1 ratio. So despite this randomization, patient will frequently appear to improve merely from being effect of placed in the clinical trial. So this is nothing but Haverthorne effect. Presently, statisticians feel the stratified balanced randomization complicates the design of the trial, and the effect of important prognostic factor can well be taken into account during the analysis. So disadvantage of block randomization is that allocation of participant may be predictable, and it will lead to selection bias, and power of calculation must demand a vast sample. And what are the types of control we usually have? First, placebo control, benchmark control, then positive control, negative control. Placebo control, where one without any expected activity against the condition under investigation. So one group will be your experimental group, other group will be your placebo group, where we will not expect anything out of them. And benchmark control, a product already is in use in a public and it is already proven to be effective, that is used here. In a positive control, accepted effective formulation is used, so it is reduced than benchmark control. Negative control, where no intervention is given. So usually to make a design easier, assign lesser patient to less favorable groups, reduce the concurrent controls and use historical control, stop accepting new subject as soon as definitive answers are available. And then adjustment of confounding factors by appropriate statistical analysis has to be done. And usually in a one group, the standard therapy asks the other group for permission for new therapy. So this is Zealand pre-randomization. Recently this is modified as double pre-randomization design. And then you have to manipulate or give an intervention. So you have done the selection of population and you have done the randomization. After randomization, you have to manipulate them or intervene them or give a treatment. So intervene the study and control group by deliberate application or withdrawal or reduction of suspected causal factor laid down by the protocol. Here the independent variable, for example drug or vaccine, is created whose effect is then determined by measuring the final outcome. And then we do a follow-up, implies the examination of subjects at definite interval of time in a standard manner with equal intensity under the same given circumstances in the same time frame for the assessment of outcome. So loss of follow-up is always inevitable. So always try to give an attrition ratio in your sample size and this can be due to either death, migration and loss of interest. And then finally assessment is done. Positive results, then benefits of experiment measures, reduce incidence or severity of disease, cost of health service can be observed in this positive result. In negative results, severity and frequency of side effects will be observed. So there will be three types of bias in this assessment. Initially the subject variation, observer's bias and bias in evaluation, and inevitably Haverton effect will be there, where the patient will feel effective when placed in a clinical trial. So to avoid this, blinding has to be done. Blinding can be done in single, double and triple blind. Blinding is nothing but where the person is not knowing what to do in the study. For example, in single blinded trial, the patient will not know what is treatment group and what is control group. In double blinded trial, where the investigator and the patient will not know. In triple blinded trial, investigator, statistician and the patient will not know what is the trial. So statistical consideration. Data is usually considered as a continuous parameter and normally distributed around the mean with observed variance, and the two types of error has to be estimated, where it is alpha and beta error or type 1 and type 2 error. Type 2 beta error is always harmful, which says that a false positive result will be accepted. And power of the study is the probability of detecting any true difference in the treatment outcome, and the primary goal of your study planning is to maximize the study power to at least by 80% or 0.80. So the power can be increased by experimental design or sample size increase. So after completion of this assessment, we have to report the clinical trial. So reporting will include your title, abstract and same IMRD format, which is introduction, method, result and comments. Introduction will define your hypothesis, method, protocol, mention whether intention to treat is there, and assessment and blinding is given, and result provide a trial profile, numerical value and rather than percentages. So this will be your format for reporting your clinical trial. Ethical considerations. Clinical trial should follow three principles in ethical consideration: beneficence, respect for rights and justice. So beneficence, which requires do good. So good should result, harm should be avoided, or benefit should justify the expected risk or harm. Respect for rights always include a free choice of subjects and protection for those diminished autonomy. And justice, which requires an equal distribution of burden and benefits. So some example for this ethical violation is Tuskegee study in Alabama on syphilis patient, where untreated syphilis patient were done. In United States, they gave this syphilis no treatment. Around 400 African were not informed of this effect of syphilis, and 100 Africans actually died. So after that, a declaration came, Nuremberg Code of 1947, where no research should proceed on human subjects without any voluntary consent, and also declaration of Helsinki in Finland 1964 by the World Medical Association and WHO, and it was revised recently in October 2008. And it says that biomedical research should be based on the scientific principle. Experimental design on humans should be formulated with a protocol, should be conducted only by qualified persons, and it cannot be carried out unless importance of objective is justified, and right of subject should be safeguarded, and predictable risk has to be mentioned. And accuracy of research should be preserved, and extra cautious taken in subjects have dependent relation with the investigator, informed consent, and subject to be told they are free of abstain or withdraw from the participation at any point of time in your study. So consent can be obtained from various participants. For example, in children's, the consent should be taken from the parents or their legal guardians. For pregnant and nursing women, therapeutic risk of fetus and neonatal has to be avoided, and research directed to include termination of pregnancy that has to be specified. Mentally ill or mentally defective persons usually never do any study that can be done on a normal patient on these patients. Agreement of immediate family members should be sorted out if necessary has to be done. Other vulnerable groups will require consideration as willingness to volunteer, and they will include the medical students, hospital patients, employees of pharmaceutical company, etc. In community-based research, consent should be obtained from the public health authority and informed community about the aim of the research, advantage and any possible hazards. So ICMR is Indian Council of Medical Research. They brought about the policy statement on ethical consideration involved in the research of human subjects in 1980, and these guidelines was reversed in 2000 as ethical guidelines for biomedical research on human subject, and the protocol should include all the following. Ethical related issues has to be from the social value, scientific design and conduct of the study, benefit risk assessment, selection of study participant, payment for the participation, protection of research participants, and community consideration, qualification of researcher and adequate assessment of study site, disclosure or declaration of potential outcomes, and plans of medical management and compensation for study related injury, review of the informed consent process. So all participants and stakeholders, including the physicians, patients and healthcare insurances as well as the NCI, other federal agencies must re-evaluate their current role and responsibilities in the clinical trial and work together to develop a more effective and efficient multidisciplinary trial system. These are my references. Thank you.